Hey everybody, welcome. Thank you all for attending our very first MCAT Mondays. So today, basically, we're just going to focus in on the Bio and Biochem Foundations of Living Systems section, otherwise known as BB in the MCAT community. You know, the goal of these MCAT Mondays is to really just pick apart a single section of the MCAT at a time, talk about, what this section really covers, what are the must-know topics for this section, and what are some of the best test taking strategies we can do for this section. And then we are also going to do a quick demo passage towards the end of this webinar. And then at the very end, we'll do live Q&A. My name is Minhal Ahmed. I graduated from Northeastern University here in Boston about 2 years ago, spent the past year in Ireland doing a master's of public health. Unfortunately, with the pandemic, that got cut short. I actually flew out of Dublin on St. Patrick's Day, which was pretty ironic. And then now I'm working back in biotech in Cambridge, and I've been tutoring with MST for about a year now. So yeah, super happy to be here. So basically, just a quick overview of what we'll cover tonight. We're going to start off with just an overview of the Biochem section. I'm just going to refer to this section as the BB section from here on out. We're going to talk about the must-know topics for this section. So, of course, if you're studying for the MCAT, it's going to be like drinking from a fire hose. There's so much to know, but we're just going to focus on what I think are things that are absolutely essential. So when I say that, I mean like you should devote enough time to actually know this like the back of your hand. There are definitely some topics on the MCAT that are higher yield than others, and we'll talk about that going forward. We're going to talk about some common mistakes to avoid, particularly with the biochem section. And then following that, we're going to talk about some test taking tips and strategies that I might advise my students on. Of course, there's lots of different ways to take the test, but these are some, tried and true tips and tricks that I think might help you tackle these passages the best way you can. And then we're going to work through a demo passage together and finally do a live Q&A. So yeah, hopefully this is a lot of use to the 90% of y'all that are just kind of getting started, and even those of you that are in the thick of it, maybe you'll come away with some test taking tips and strategies that might elevate your game a little bit. So who we are here at MST, we are basically just a bunch of people who love teaching and love seeing students reach their potential and succeed. So at MST, we do a lot of one-on-one online tutoring all the way across the, medical school pipeline. So from pre-med to all the way through residency. We tutor for MCAT and for USMLE, step 1, step 2. MST's been around for 14 years and has, really put a lot of passion and effort into seeing their students succeed. And we've got med students, residents, and attendings who, just love what they do, love what they, love the chance to kind of interact with people who they see as future colleagues, and just love to teach. And so it's a really great community to be a part of, and I've really enjoyed my year with them as well. What we're going over on this slide is just an overview of what the biochem section entails. For a lot of you, this might be stuff you already know, so we won't spend too much time on it. And Faith is going to put up this poll here about what your biggest concerns are for this section. So this is just something I want to get a feel for, from students. There's typically 3 big areas that are the most concerning. It's either the timing, the content, or something about the test taking. So, and while you're all doing that, I'll just go through the section. So the biochem section is actually the 3rd section in the 4-part MCAT exam. So the MCAT has 4 sections. Biochem comes 3rd after Chem Phys and CARS, which is the critical analysis and reasoning skills section, and before the final section, which is the psych-soc section. Like the other science sections, it is 59 passage-based and discrete questions, and it's about 44 passage-based questions and about 15 discrete questions typically. And you have 95 minutes to do so. So what this sort of breaks down into timing-wise is that people say a good rule of thumb is to spend 8 minutes per passage, including reading through the passage and also answering those questions. But subjects tested on this section are it's the biochem section. So what do you expect? We've got 25% of the questions that you'll see here are going to be from first semester biochem. 65% are going to be from intro bio, and then about 10% are going to come from gen chem and orgo. So even though it's the biochem section, you're still going to see a little bit of chemistry, but the big bulk is going to be biochem and bio, as the name sort of gives away. The skills tested on this section, and so the way the MCAT's designed is they're looking to test not only like whether you know these very foundational scientific principles and concepts, but also they're testing your scientific reasoning skills. And so the skills that are broken down here for the biochem section are knowledge of scientific principles, scientific reasoning and problem solving, reasoning about the design and execution, research, and database statistical reasoning. And so you'll notice here, when you think about the MCAT, a lot of times students think about just in terms of, the vast amount of knowledge they need to learn. And that's typically what kind of provokes a lot of the anxiety and the dread about the MCAT, is just all this stuff you need to know. And I mean, looking at this poll, and I'm not surprised by this poll because, from, the tons of students I've worked with, it's always the biggest concern is the content. But what I want to show to you with this slide here and this breakdown straight from the AMC is that almost 70% of the skills that are tested on this section, and actually a lot of the science sections on the MCAT, have to do with reasoning. So only 35% of the skills tested on this section are knowledge of scientific principles. That's like your bare bones facts, right? Like whether you know how the electron transport chain works, blah, blah, blah. But almost 70% comes from actually working with the passage that you're given and working with the figures and tables that are presented and actually critically thinking about them and analyzing that data. So in some ways that can be sort of, it depends on the kind of student you are. That might be reassuring or that might be a little scary. I hope you all, by the end of this session today, will walk away feeling reassured, because to me what this shows you is that you can really do very well on the MCAT with minimal content under your belt. And I think that's a big mistake that a lot of students make, is spending so much time in the books and not enough time in the practice books, not doing enough, passage-based practice, because so much of what is tested is really your test-taking skills and your reasoning skills. And that really comes through good test-taking strategy and really critically thinking about how you're analyzing these passages. I see Brittany's got a quick question, so I'll just see if I can answer that right now. So in respect to the biochem content of the biochem section, how in-depth does or can the MCAT get? For instance, amino acids and structures, reactions, etc. So that's a great question. And I think a good rule of thumb to think about is that the MCAT, even though it tests a wide range of content, wide range of subjects, the phrase goes, it's a mile wide, an inch deep. So, as you can see here, this is about first semester biochem and introductory biology. This isn't, some graduate level stuff. So to simply answer your question, it does not go into, much deeper depth than your college courses went to. I would say it's actually probably more superficial than a lot of folks' college courses went. But you'll see that in whatever review books you choose to use, you'll see kind of the level of depth they go into. And we'll talk a little bit more about how any of the, passages might go deeper than just the fundamentals. But what you'll realize is that doesn't mean you need to have known that ahead of time. The passage is just going to kind of present that information to you. So that was a kind of long-winded answer, but I would say it's very stick to the fundamentals for the MCAT. It's very much a lot simpler than I think people might construe the exam to be. Awesome. Okay, so we'll keep it moving. So this is just a breakdown of the biochem section for those who might not be familiar, and for those who might be familiar but not familiar about the skills tested, just to put in perspective how important the test taking aspect is. Okay, so I guess kind of building off of Brittany's question, right? What are some of those must-know topics? And you'll get a sense for how deep the MCAT goes through these topics. So I'll preface this slide by kind of saying that this is by no means an exhaustive list. It's not even an exhaustive list of all the must-know topics. But if I had to give you 3 things that I think are the most high yield for the biochem section, that if you just, if you had to memorize any 3 things or really deeply learn any 3 things, it would be these 3 categories. And we'll go through them one by one. So the far and away front runner for this is going to be all the amino acids. So that I can guarantee you are going to see several questions that rely on deep knowledge of the amino acids. And I know that can be daunting at first, but I promise you with practice and repetition and a little creativity, you can absolutely, learn everything you need to know about the amino acids. So what about the amino acids do you really need to, learn? Well, the 1-letter and 3-letter abbreviations and the structure are actually a must. You need to be familiar with what all these amino acids are called and how they're referred to in those abbreviations, but also their structure. And you'll see some patterns as you go through those amino acids that'll make that easier. Unfortunately, that's kind of outside the scope of today's section. But if anyone has questions about tips about any of this stuff, feel free to ask them towards in the Q&A session. But most importantly, you need to know the properties of these amino acids. And these break up nicely into, acidic amino acids, basic amino acids, polar amino acids, non-polar amino acids. You want to kind of break up the amino acids and know which fall into those really important categories, because, with an understanding of that, you'll be able to answer so many questions that sort of rely on an understanding of, well, yeah, the polar amino acids are going to be found on the exterior of a protein and the non-polar amino acids are going to be found on the interior, right? And so you need to know which amino acids are which there. So that's beating a dead horse, but absolutely know all the amino acids. I recommend Amino Acid Quiz on the App Store. I'm not sure if it's on Android, but I use that app. It was awesome. Makes it kind of like a game to learn those amino acids. The other thing that is a big source of dread, which I totally, I feel for everyone taking the MCAT. Hey, I went through it. The metabolic pathways can be daunting, right? And I think that you absolutely should know these metabolic pathways that we've listed, and I'd sort of rank them in the order that they appear in terms of importance. But notice how I paired them up. What you want to understand when you're going about learning these metabolic pathways is try not to understand them or learn them at the granular level and don't silo them. These pathways are all part of, homeostasis in a way. They're very interrelated. And so, glycolysis, the Krebs cycle, fermentation, and gluconeogenesis all go together because they're all related to, glucose metabolism, right? Or glucose anabolism. And ATP synthesis, I kind of put aside just because I didn't want it to wrap, but it's also kind of related to this. It's related to the Krebs cycle. So that's super important, and try to learn these metabolic pathways in relation to each other. Know that, fermentation and gluconeogenesis might be kind of opposites, or vice versa. Same thing with glycogenesis and glycogenolysis, right? One's just the synthesis of glycogen, one's just the breakdown. And if you understand their interrelation and the key enzymes involved, it'll be a lot easier. Same thing with fatty acid synthesis, beta oxidation, and pentose phosphate pathway. So I won't spend too much time there, but I will say when you're learning these metabolic pathways, pay special attention to the rate limiting enzymes in these pathways. There's so much to know. Pay attention to those key enzymes that are the regulators of this whole process. And each of these processes has a rate limiting enzyme. And just make a special note to, if you had to prioritize what you learn, make sure that's at the top of the list. And then finally, this is really related to the bio part of the biochem section. You got to know the main organ systems. And all of them are fair game, but of the three that I think are the highest yield, I would say the nervous system, the endocrine system, and the renal system. So, understanding kidney biology and nephron biology is really important. And again, I'll talk about this a little bit later, but the focus on the big picture here. Really pay attention to the relationships. How does, if you tweak one thing, how does that affect another thing? How does the parasympathetic nervous system relate to the sympathetic nervous system? And don't study anything in isolation is basically my point, and that goes for all of these. Okay, cool. So those are just some must-know topics. The other topics that sort of made the short list for this were different biotech, or what's the right word? Different kind of experimental methodologies for molecular biology. So, Western blots, Southern blots, and Northern blots are a good one. I'm just going to quickly go over this tip here because it can be kind of hard to remember which one's which. A good acronym or mnemonic I like to use is SNOWDROP. So I'm not sure if I'll be able to write it here, and I'm actually probably not going to attempt it live so I don't embarrass myself. But if you write down vertically SNOWDROP, so it's SNOWDROP. If you have a piece of paper next to you, you can do that. You'll see the S and the D line up. That's Southern blots are for DNA. The N and the R will line up, and that means Northern blot is tied with RNA, and then finally the W and the P are lined up, and that's because Western blot is tied with protein. And yeah, that was a life-changing mnemonic that I learned. I can't take credit for it. Don't know who came up with it. Definitely should be tapped for the Nobel or something because it's huge. Cool. So moving forward, so that's just kind of like a high level overview. These, if the 80-20 rule, 20% of all the things you learn have 80% of the biggest impact. I would say these all fall into that high impact category. Cool. All right. So I hate to kind of frame this negatively, but sometimes there's some common mistakes that are very prevalent in the pre-med community taking the MCAT. And they're all understandable, but I want to make sure that you're able to fix these errors early on in your prep and really be on the lookout for them, when you go forward with practicing. So some common mistakes to avoid are skimming the passage. I think that even for science sections, I think students sometimes feel like you might be able to just get by with skimming the passage because it's not like it's a CARS passage. You don't need to understand this really arcane language about architecture. But actually, you should try to read every passage really carefully to understand the main idea. And this is because, going back to the earlier slide where I showed you the breakdown of skills tested on this section, it's really about your reasoning skills, right? And in order to reason well within a passage, you need to have a very strong grasp of the passage. So I am a firm believer in slow and steady wins the race. When it comes to the MCAT, I read all the passages on the MCAT very slowly. Like I pretty much pause after every sentence to do a little brain check to make sure I understood it and say it in my own words, and then read the next sentence. And I know it can seem a little nerve-wracking at first because you're like, you're timed, you're on the clock, but I promise that investment actually pays off in the questions. You'll be able to kind of see through a lot of the questions that are thrown at you and understand how to go about them. And that's something that can really build with time. But I'd say get in the habit of reading your passages really carefully and trying to grasp the main idea rather than just kind of speeding through. And absolutely do not look at the questions before reading the passage in its entirety. The next common mistake to avoid is overlooking the titles of figures and tables. And that might be a visual thing, right? You see a big fancy graph and a lot of you are probably STEM people, so your eyes go to that. And you don't think the title is that important, but titles give away the main idea of any figure and table. They can really show you what the independent and dependent variable are. So pay attention to the titles for sure, every single time. This little, of course, because acronyms are the name of the game here, use the TAP method when analyzing figures and tables. What the TAP method is title, axes, pattern. So we'll go through this in the demo passage, but whenever you see a figure or table, read the title, then go, look at the x-axis and the y-axis, make sure you understand what the units are, how they relate to the independent and dependent variable, and then start by looking for patterns, right? And don't, when you're analyzing a graph, don't just take it all in at once. You don't want to parallel process that thing. You want to go step by step, focus on one single line or one single, group of bars, and then extrapolate further and further. And that's easier to see when we go through a practice passage. Next thing is using too much outside knowledge. This is a big common mistake. And I think it comes from the sense that, people tend to focus on the content that's needed for the MCAT. Like, you need to know all these things. You need to be an encyclopedia of biological knowledge. But that's absolutely not what the MCAT is really about. The MCAT is about understanding the fundamentals, fundamental concepts, knowing that, it goes DNA, RNA, protein, knowing that polar and non-polar don't really mix, things like that. And then applying them in the context of these complex passages, right? Even when you see a question that seems very deep, seems very complex, sometimes students kind of fall back on some outside knowledge because they think that this question is testing this really, arcane knowledge that not a lot of people have. The truth is that almost all the questions are tied very neatly with the passages, and you'll get a sense of this as you do more and more practice. But what I would, the piece of advice I would give you to steer you in that direction is to really make an active effort to stick to the theme and the scope of the passage to guide your reasoning. Try not to use outside knowledge if it's not a fundamental fact, right? Or it's not a fundamental principle that would definitely be covered in your review books, right? Those are kind of high level. Don't ever think that, you're expected to know something that was really specific that you learned in one of your high level advanced coursework. That's almost never going to be, even if it is right in that scenario, that's not the path that the AMC expected you to take to get to that answer. The path is almost always through the passage, and there's an easier way there. So try not to use too much outside knowledge. Moving forward, not doing enough passage-based practice questions. This again, you're getting hopefully a theme here. It's not all about, memorizing content. It is all about practice, and it is all about reasoning and getting better at that. And so big mistake that more on the prep side is not doing enough passage-based practice questions. So I think that, in the students I work with, I try to make practice actually the majority of the study schedule. So I go for like a 70-30, 60-40 breakdown of practice versus just kind of content review. And definitely in the later ends of the MCAT prep kind of window, you want to go pretty much all in on practice, because that is content review when you review those. And there's a whole lot there on how to properly review your practice, and maybe we'll get to that in the questions and answers, but that's kind of outside the scope of today's session. And then finally, neglecting the big picture. This goes well with the prep. I know a vast majority of you guys are just starting off in your prep. Really try to focus less on the minutia. You don't need to know, I'm not going to say you don't need to know anything, but I will say that you can get by knowing the fundamentals really well. You can score really well just by knowing the fundamentals. So when I say fundamentals, I mean those foundational principles those foundational relationships, understanding how, you know, what the levers are that push gluconeogenesis one way and fermentation another, right? Like, why does our heart rate increase when we're exercising? Oh, it's probably to, you know, pump blood to our muscles and exhale CO2 that's being generated. Like, I'll try to focus on those relationships more than learning things in isolation. And that'll really help you when you're getting ready for the test. I don't know if that example with the exercise made sense, but hopefully you're getting my drift here. Okay, cool. So those were a little bit about, that was a little bit about some test taking tips and strategies, and I'm just gonna check my time here. Cool, so we're about halfway through, perfect. But some more on test taking tips and strategies for this section are fundamentally, and this goes for all science passages, actually all passages, do not over highlight because you think that it's, you know, the best way to engage with the passage. What tends to happen is that you kind of make it harder for you to go back to the passage, which you definitely need to do when you're answering questions, and pick out the important information. Because when you're reading the passage, you don't really know what the pertinent bits of info are all the time. So really limit your highlighting in the biochem section to just the central hypothesis or the goal of whatever experiment is being described. There will almost always be an experiment that's, you know, the heart of the passage you're looking at. So and we'll do this in our demo passage, but really limit your highlighting to the central hypothesis and goal of the experiment. One thing I'd add there that is not written is highlight the independent variable and the dependent variable that the scientists in the passage are investigating. Other testing tips and strategies, this is more kind of getting into the nitty gritty here, but what I really recommend when you're going through the section, like what does that actually look like? I always read the passage slow and steady first, all the way through before looking at the questions at all. If there's figures throughout the passage, I only read the figure titles during that read through. And that's because the figure titles basically tell you exactly what that figure is supposed to tell you. Figure titles in scientific literature, thankfully, are very informative. They're designed to tell you exactly what is going on here. So the reason I do that is because I'll be able to read through the passage, know that, okay, figure 1 is showing me the relationship between X and Y, and table 2 is showing me this breakdown of that. But I don't need to spend so much time during the read through to, you know, to really dive into the details of those graphs, because there's a chance none of the questions will actually even use that graph, right? And that does happen. And so because it's such a time intensive thing and brain intensive thing to analyze a figure, I only go back and do that when a question specifically calls for it. And it may not, whether it's direct or indirect, right? So that's a tip that might save on time for students. Okay, and then next tip is don't get lost in the sauce. So really what I'm trying to say there is just don't be afraid by all these, you know, 20 syllable words that you're reading in the biochem section. That definitely tends to happen also in the chem phys section. Don't let it scare you. They're just names. So get in the habit of not even reading those long drawn out words of, you know, enzymes or whatever. Just sort of simplify it to an initial or something that, you know, flows for you, and keep it moving. Simplify all the jargon. A lot of these passages are actually just jargon. And if you mentally sort of figure out a way to work through it, and when I say figure out a way, I mean that comes with practice and doing lots of these passages over and over again. You're going to be able to kind of read this through by like, oh yeah, blah, 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 a thingamabob does this and that. And then you won't, you know, kind of be daunted by those really complicated words. And that sounds super simple, but at least in my experience working with students, it actually goes a long way in just kind of confidence levels and how you go about a passage. It's not important what those names are, not always. The only exception I'll make is the class of enzymes can be important. So whether something is a phosphatase or a kinase or a, hydrolase, those are things that you'll learn in your content prep. And that little bit of a long drawn out enzyme name can be useful to actually understand what it does, right? Otherwise, not so much. And also, yes, this PowerPoint will be provided after. The session will be recorded, so you'll be able to see it there. Next, identify the independent and dependent variables. We've already talked about this, so I'm not gonna, harp on it too much, but that's super important because a lot of this is dealing with, understanding experimental design and just to understand the relationships in a passage. You wanna make a point to make those independent and dependent variables really clear to you. So I would highlight them or write them down on your note sheet. And then finally, this is the big kind of takeaway, I hope, from this session. This test taking strategy, I think, will be the most impactful in kind of changing your game on how you approach the MCAT. And that's making simple flowcharts of relationships and pathways. And I'll show you an example of exactly what I mean by that. Sometimes the word flowchart can scare students or whatever. It feels like a lot of extra work you want to do, but I am so opposed to extensive note taking or drawing out these schematics on your test day. I know you're going to be nervous. I know you want to do things that are, really efficient. When I say simple flowcharts of relationships and pathways, I mean literally writing down initials, up arrow leads to, this down arrow. And that's it. That's all I mean by a flowchart. And I'll show you exactly what that looks like. But getting in the habit of converting these long sentences that are sometimes written with poor grammar into these really simple flowcharts of relationships will be super useful for you, because almost all the questions come down to an understanding of those relationships and pathways. That's really what you're trying to extract from the passage that you're reading. Because a lot of these passages, as you'll see, are, adapted from the literature. And so they'll have a kind of pattern to the way they're structured. They're going to start off with some background, then they're going to, give the rationale for a study. They're going to, and in the background, they'll kind of introduce these relationships. And then the scientists might focus in on one independent variable and see how it affects another dependent variable that was kind of hinted at before. And then there you go, right? It's all about those relationships and pathways. So making those simple flowcharts can go a really long way and just help you see passages a lot more clearly. Okay, so all of that being said, it's one thing to kind of hear about them. It's another thing to put them into practice. So I know that, a lot of people learn just by practice. And so that's what we'll do here. I purposely chose a really short passage. This is, from Khan Academy, which is a free resource for, practice MCAT passages. And I'm going to read through it, go through how I would highlight and make those flow charts for this passage. And then we're going to, do 2 questions together. And then after that, we'll have the Q&A. And I think we're doing all right on time. So, alrighty. So I'm just going to read this out loud exactly how I'd read it on a test day. Okay. So follicular lymphoma. So already I'm just rethinking that as like FL is a blood cancer where B cells have an unusually long lifespan, much longer than that of normal B cells, which can be as short as a few days and as long as 2 months. Okay. So FL, B cells have a really long lifespan. That's all I got out of that. These longer living B cells cluster in the lymph nodes and clog the lymphatic system. Through karyotyping, which lines up all the chromosomes and compares homologous chromosomes, scientists have observed that chromosomes 14 and 18 appear different in the longer living B cells than they do in normal B cells. So then I'd be thinking, okay, these long-living B cells have something different about chromosome 14 and 18 than normal B cells. Moving on, they concluded that there was a translocation between chromosome 14 and 18. It has also been observed that these B cells with translocations express a protein identified as BCL2 in excess. Okay, so recapping, karyotype was different, right, between normal B cells and longer living B cells. The karyotype was different because of a translocation between chromosome 14 and 18. And that's a little bit of content that you might need to bring to the passage to understand, but a translocation, hopefully some of you might know, is when a piece of a chromosome or a DNA strand, moves to another chromosome. And really simply, that's all, right? So that's why this karyotype was different. So, and these B cells express BCL2 in excess. So I don't care what BCL2 is, I just know that they express it in excess. So FL is a slow progressing cancer, and the B cells inside an individual with FL do not divide any faster than normal B cells. That's interesting, right? Even though these B cells in FL have a really long lifespan, they're telling us that those B cells don't divide any faster than normal B cells. So they live longer, but they don't divide faster. Okay. It was hypothesized, so there's the key here, there's the hypothesis and goal in every passage. It was hypothesized that the overexpression of BCL2 might be related to the extended lifespan of these B cells. Normally, B cells are terminated by apoptosis, which is not observed or triggered as quickly in these longer living B cells. So apoptosis, it doesn't happen the same in these longer living B cells. That's interesting, right? To test this, researchers created an antisense, see, I'm not even gonna read that word, simplify the jargon, don't get lost in the sauce. So antisense PS ODN of the BCL2 gene. I just said ODN because I'm familiar with this term, but again, if this is a scary term to you, just see that it's a nucleotide and move on, right? It doesn't matter. So to test this, researchers created an antisense PS ODN, or a nucleotide, of the BCL2 gene and observed cell density over time when this antisense PS ODN was introduced to colonies of B cells that express BCL2 in excess. So even as I'm reading that, I can tell that was a mouthful, right? But let's break this down. Basically, researchers created some antisense strand of this gene, and they looked at cell density over time when they added this antisense strand into colonies of B cells, right? Colonies of B cells that express BCL2 in excess. So you want to chunk these sentences, right? Introduced to colonies of B cells that express BCL2 in excess. By that, they just mean those longer living B cells, right, from FL, because those are the B cells that have BCL2 in excess, right? So they're looking at those FL cancer cells. And they're looking at those cells after they added this antisense strand of the BCL2 gene there. Cool. So a phosphorothioate oligodeoxynucleotide is similar to a phosphodiester oligodeoxynucleotide, except an oxygen atom has been replaced by a sulfur atom. So I don't care about that at all because that doesn't really add anything to me. Like maybe that'll get called up in a question, I know where to go, but it doesn't, it doesn't jive really with the main idea of this passage, which is about B cells and their lifespan and why do they have these longer lifespans, right? And then so to move on, there's just a little bit more in that passage. And it says, to make sure the results from the antisense PS nucleotide experiment were accurate, the researchers also observed colonies of B cells exposed to a BCL2 PS sense strand and colonies of B cells exposed to saline solution. Okay, so they just introduced two other groups to us. The results are shown in figure 2. And yikes, okay, so this is a Khan Academy passage. Doesn't have a figure title, which, all the MCAT passages will have a figure title. But if this had a figure title, I would just read the figure title and move on, right? And the title would just be relationship of, the effect of antisense nucleotides on B cell density over time. That's it. And I'd know that this is where I would go to look at that relationship. Okay. So moving forward, right, we just did a quick read through of that passage. I didn't actually make those those pathways yet, but I'll show you how useful they are. So the first question we see here is, what is potentially not a consequence of early stages of follicular lymphoma, right, of FL? So which of these is not a consequence of early stages of FL? So I'm going to tie this back to those common mistakes I talked about, right? Some students might read this and immediately go back to their content, right? They'll be like, oh crap, I don't think I read about follicular lymphoma, or like, what's lymphoma? Okay, lymph, it's white blood cells. Like, what does it have to do, right? Save yourself the effort. This is clearly something that is not what you needed to know. You don't need to go into this depth in your biology review to know exactly what all the different kinds of lymphomas are about, right? So because you know that this seems a little bit out of the scope of what the MCAT typically tests in the sense of the fundamentals, this is probably something that you want to use the passage to answer, right? And so in the next slide, I'm going to go back to the background paragraph where they talk about follicular lymphoma, right? And I've already made these flow charts that I referenced earlier. And so basically I simplified this entire paragraph just down to these two things, right? Let's look at the sentence first, and then we'll look at where this came from. So follicular lymphoma is a blood cancer where B cells have an unusually long lifespan, much longer than that of normal B cells, which can be as short as whatever, blah, blah, blah. These longer living B cells cluster in the lymph nodes and clog the lymphatic system. Okay, so I'm going to stop there. So we learned a lot there, right? We learned that FL happens when the B cell lifespan is, longer, which leads to lymphs being clogged, right? And that's the relationship. FL is B cell lifespans increase, and then clogged lymphs increase. And then if I go on through the karyotyping thing, I'll see that, they saw that in those longer living B cells, there was this translocation of the chromosomes, which lead to an excess of protein expression of BCL2. So that adds another piece to the story that we can simplify, which is that BCL2, when that's, expressed in excess, that also leads to B cell lifespan being increased. So, so now we have a good, bird's eye view of what follicular lymphoma looks like. I'm just going to focus on this first top line, right? FL equals B cell lifespan increases, which leads to clogged lymphs increasing. So going back to this question, what is potentially not a consequence of early stages of follicular lymphoma? Well, we know that follicular lymphoma basically leads to clogged lymphs. So which of these would not be a consequence of clogged lymphs? Okay, enlarged lymph nodes definitely sounds exactly like what would happen after clogged lymphs, right? So it's so easy when you have this flow chart. Fluid buildup around lymph nodes would definitely happen when you have these clogged lymphs. So that's out too. Lowered immune response versus easy bleeding and bruising. So here it might be a toss-up, right? It might not be as obvious that when you have clogged lymphs, it would affect your immune response. But hopefully, if going back to how you want to focus on the big picture when you're studying, hopefully if you focused on the big picture enough, you know that the lymphatic system and the lymph nodes are really about, your white blood cells and your immune cells, right? So which of these has more to do with your lymph nodes, just based off those really, high level basic associations? You'd know that clogged lymph nodes would most likely affect your immune response, right? More than your bleeding and bruising. So you can eliminate that too. So which of the following would potentially not be a consequence of early stages of FL? Easy bleeding and bruising is the right answer here. And we got that really simply, by having drawn out a flow chart that showed us FL is really just leads to clogged lymphs, right? Awesome. And I actually don't have the chat open, so I don't know if folks are asking questions about that, but we can go through those later. When you make a flow chart, do you make them as you read the passage or after you finish reading the passage? That's a great question. I like to do it as I'm reading the passage. I think that it's just more efficient time-wise. But one caveat I'll add here is that, this is just how I'm presenting a potential strategy to you. And I'm kind of giving you tips and tricks and, you know, little nuggets of information. Feel free to put them together, tweak them, not use some stuff, use the other, as much as you please. I definitely think that everyone has a different way of thinking, different way that works for them. So, you know, when you're going about your practice, you know, experiment, see what works better for you. What I typically do is make those flowcharts as I'm reading. So just for the sake of this passage, this slideshow, I did it afterwards, but normally when I'd read this sentence and this next sentence, I would write out that first flowchart. Okay. Next question. So what role does the overexpression of BCL2 play in the development of follicular lymphoma? Okay. So what role does this overexpression of BCL2 play in this development of this cancer, right? So going back, we saw that BCL2 being overexpressed leads to an increased lifespan of B cells, which is basically what causes this cancer, right? So already just using that pathway that we brought out, we would hypothesize that an overexpression somehow leads to something that, you know, would prolong these cells' lives. And, you know, at this level, I'm talking assuming that you know these biological concepts, but you might not. And that might be stuff that you focus on in your review. But I'm assuming, you know, I'm assuming knowledge here that apoptosis is, you know, programmed cell death that happens that would necessarily be the opposite of prolonged lifespan of cells, right? If you have an apoptotic cell, you're not going to have a super long life, a long living cell, right? So if based on our pathway, BCL2 increases, overexpression of BCL2 leads to longer B cell lifespan, we can be pretty sure that BCL2 is not an apoptotic protein, right? Because that wouldn't fit with the pathway. That would actually, overexpression of an apoptotic signaling protein would lead to, you know, cell death. So the next thing is an anti-apoptotic signaling protein. That makes sense, but we're not sure. That fits with, you know, a longer lifespan. That fits with our pathway. BCL2 is a transcription regulating protein, you know, potentially, maybe if it increases transcription of certain, you know, key enzymes, it could prolong the life of the cell. Or if it's a receptor for a growth hormone, again, right? Increased receptors for growth hormones might lead to longer life. But I'll actually pause there and say one other thing I could have added here is that increased BCL2 does not lead to, you know, more division of these cells. So maybe I would actually X this out because that doesn't fit with the knowledge that we're given, but I'll skip that. What I really wanted to do there was just use that flow chart to X out choice A, right? Because we can be clear about that. We can't use it to really narrow down these three. What we do here, oh, and I'm sorry, this is a little bit out of order here, but basically this is just to give you a sense for how I would highlight this passage, right? This is the entire amount of highlighting I would do on this passage, and I just highlighted the hypothesis, which is that the overexpression of BCL2 might be related to the extended lifespan of these B cells, right? And that's tying back to the question. And then to test this, researchers created this thing and observed cell density over time. I highlighted that because that's the dependent variable when this antisense PS ODN was introduced, right? That's the independent variable that they're introducing. So I just highlight the hypothesis, dependent and independent variable. That's it. And right, and so going back to the figure, right? Because I just saw that the question that I'm trying to answer has to do with the figure, right? What role does the overexpression of BCL2 play in the development of follicular lymphoma? Well, let's see what happens in this figure, right? And we'll use the TAP method. Unfortunately, there's no title here, but that would be the T. Next thing is axes, right? Axes on the x-axis, we see time in days. On the y-axis, we see cell density in 1,000,000 cells per mil. Okay, so we have the dependent variable on the y-axis, and then the independent variable are going to be in these different lines here, right? We've got antisense is the one with triangles, sense nucleotide is the one with squares, and then the saline control group is the circles, right? So let's do one at a time, right? This is the pattern part of the TAP method. So I like to start with the controls always. So the control we see here, right? This is just the reference group. We see cell density increases between 0 and 4 days. It goes up, and then it sort of starts to go down gradually afterwards. Okay, so that's when we don't do anything to the cells. Let's look at the antisense, right? This is when we're adding something that is antisense to the BCL2 gene. We should know based on our biological knowledge that that would kind of cancel out the BCL2 gene. It would act against its expression. So here we see that for this triangle one, it starts off the same as the control group, but then we don't nearly see the same amount of cell growth, and it goes all the way down, right? So what does that tell us? That tells us when we take out the BCL2 gene, or we silence the BCL2 gene expression, you get less cell growth, right? And so you'd be able to deduce from that what BCL2 is used for, right? Like it might be doing something that's keeping the cells from dying, right? Because when you block it, the cells die. And then just the positive control group, when they add more BCL2, right, in the sense case, you see that it's basically like the control group, except you get a little bit more, cell growth, and then it levels off the same. And that again fits our hypothesis about BCL2, that it's something that would actually, stop cell death. So going back to this question, the only thing here that would stop cell death, right, when it's overexpressed is anti-apoptotic signaling protein. And that might have been obvious to some of you beforehand, but we really can concretely come to that conclusion based on the figure that we just analyzed there, right? Because when you have a lot of an anti-apoptotic signaling protein, you're not going to see apoptosis, and therefore you're going to see longer living cells. Yeah, I won't beat a dead horse there. But importantly, you wouldn't see that for these other two cases. Those wouldn't lead to the patterns we saw in the figure. Okay, so cool. We are just about wrapping up here. So that was our demo passage, was just 2 questions. And that's basically the gist of everything that I wanted to cover today. I hope that was helpful. We still have the live Q&A, so I'm definitely sticking around. But hopefully you got to see some of those tips and tricks and strategies in action there, and just how I go through a passage. And maybe you saw stuff you liked, you didn't like, but we can talk about that later. And so finally, just a little bit more about MST, what we do here. Really quickly, MST is founded on this one-on-one personalized tutoring. The reason I love tutoring with MST is because, I just love working with my peers and kind of forming those friendships and relationships with people that share my same interests and want to go into the same field I do. It's really easy to connect. And I love being able to build those relationships and kind of get my students to, reach their goals. And I can absolutely say that everyone at MST shares in that kind of, passion and goal. So if it's something that you're interested in, for, as you prep for the MCAT, feel free to, look into MST for, as a resource for you. And yeah, and this was just the first of our MCAT Mondays. We're going to do one next week as well for the CAR section, and the following weeks for the Chem Phys and Psych Soc section. So feel free to add that to your calendar. It's going to be at the same time, 7:30 p.m. Eastern, and you'll be able to, jump in. We'll go through a CAR specific, test taking strategy and do a passage together, and have the same Q&A afterwards. But with that, thank you all for your attention. And yeah, I'm happy to take any questions that anyone has. How long do you recommend a person? Thanks, Brittany, for your question. How long do you recommend a person who has a general understanding of BCP content study for the MCAT? So that's an interesting question, right? And without too much information, it's hard for me to, really give a concrete answer. But when I plan out just for a generic student, I typically say for a 3-month study schedule, spend the first month going through the review books and doing, practice questions alongside about, 30% of the time with like UWorld or something. And then spend the next 2 months really diving into more just practice questions, right? So I think if you feel like you have a general understanding of BCP content, definitely go through the review books, but spend most of your time doing practice questions using resources like UWorld, whether you like Kaplan or Princeton Review, Khan Academy is a free resource. And then finally, of course, all the AMC material, really dive into that. And that'll be kind of how you fill in your content gaps when you review those questions. So practice is never just practice, it's also studying. Okay, hopefully that answers your question, Brittany. And then Roseanne, you had a follow-up. So basically we study on our own using review books and then go over it with a tutor. Okay, interesting question. Yeah, so the way one-on-one tutoring works with MST, if anyone else is curious, it is very personalized. So for example, when I meet with a student for the very first time, I hear them out on their needs, where they're at. We figure out how comfortable they feel, how far along in their prep they are. And then we work to develop a study plan, right? I build out a very personalized, calendar as detailed as my student wants it to be. And then that's sort of the structure for their prep. And we'll meet, anywhere from 1 to 3 times a week, really depending on the student, mostly for lessons. And then lessons similar to this, but obviously more detailed and more personalized, and to go over practice questions. And so they can ask me questions and work through passages together. So that's typically how tutoring works. We definitely won't, like read a review book together. I think it would be, I definitely have students who ask questions from their review and we go through them that way, but it's kind of like a flipped classroom type thing. Hopefully that answers the question. And then Sina, I hope I'm pronouncing your name right. People pronounce my name wrong all the time, so I hate when that happens. Hopefully I'm not pronouncing it wrong. But how long do you recommend studying for the MCAT to avoid burnout? I would say, it's tough. Everyone's different. I try to tell my students to focus for that 3-month sweet spot, maybe 4 months. Everyone's schedules are different, but I really think, 3 to 4 months is perfect. The number that you want to shoot for is 300 hours total for studying, is like on average what students do. So if you have a test date in mind, the way I typically work is, take those 300 hours, divide it by the amount of weeks you have until your test day, and then you'll get a number, right? Maybe it comes out to, you have 300 hours, you're taking it in 100 days. And so that's, 3 hours per day, right, that you need to study for the MCAT. Is that something that's feasible for you? If it's not, then push the MCAT further back. And that's how you would develop that sort of timeline, if that makes sense. But I shoot for that 3 to 4 month timeline. So is there an area that you consistently see students struggle with for the biochem section? If so, what would you recommend students do to make sure they really get that topic down? That's a great question. So I think that honestly, the biggest thing I see students struggle with is not with the content. I think everyone I work with is super smart and driven and able to dive into the materials, and they have a really strong grasp of the fundamentals. Where they struggle with, and this kind of doesn't apply to a particular topic, but rather the entire section, is being able to draw the line between, this is outside knowledge, and using the passage to guide you and take the easy way out, right? So that question we went over with the follicular lymphoma, students who've studied a lot are going to default to thinking that it's something they should have already know, right? And I think the challenging thing is to really pay attention and really get in the habit of always going back to the passage and using the passage to help you. In that way, ironically, it ends up being an easier test, and you have to know less when you learn how to use the passage to guide you. You know, there's a brute force approach to answer these questions, and then there's a lock and key approach. If you find kind of the breadcrumb trail that the question is leading you on and the passage is kind of built for you, everything will really just rely on the fundamentals. And nothing really has to do with you knowing super arcane things. If there are no other questions, hopefully it was helpful. I really like doing these things just because, you know, they're a way for people who are just starting out, you know, planning out their prep to kind of get an idea of how a section works, really tangible things you can take away to your own prep, and hopefully feel a little bit more comfortable about how the MCAT, you know, operates and how you can do really well on it. Because I absolutely think that, you know, you don't need to be some genius, some special person to score, you know, in the 99th percentile on the MCAT. I think that's within reach of just about everyone. So it's really about kind of figuring out the test, because it's a little bit different than, it is a lot of bit different than a lot of the tests that college students have taken. All right. Thank you all, everyone, so much for joining in. Hopefully I'll see some of you next week for the CARS MCAT Mondays. You know, be well, and see you guys soon. Thanks.